So what is cell cycle?
uh, division, cell division, data.
Cell cycle is the various events which take place for the cell to divide. Yeah. Now growth and reproduction of all organism depends on cell division and cell enlargement. We can grow in two ways. Either the cell should divide or the cell should become large. The mechanism of division and multiplication of cell together constitute cell reproduction. Okay. Don't write these points now. Then second is, it is the life period of a cell during which cell synthesizes DNA replication, grows and divides into two daughter cells. Every cell will divide into two daughter cells. Cell growth, that is cytoplasm, will increase, is a continuous process. Cell will continue to grow, but division, it will take certain time. Then it will continue to grow, divide. Growth is continuous. DNA synthesis only occurs at a specific stage. Which stage it is? DNA synthesis S phase. S phase of the interphase. Okay. Duration of cell cycle varies in each organism and each cell type. Different organism cell cycle will be different, and different cells also, the time for cell to divide will be different. For example, in human, it is about 24 hours. One cell will divide into two in 24 hours. In yeast, it is around 90 minutes. And in bacteria, it is 20 minutes. Okay. Then cell cycle has two basic phases, interphase and M phase. So these are the phases, interphase and M phase. Interphase, you can see the full red, and M phase is the smallest phase. M phase stands for mitotic phase. Okay. Now, during interphase, it is the phase between two successive M phase. Between two M phase, there will be one phase called as interphase. Between two M phase, that is between two cell division phase. M phase is where the cell actually divides. There will be one interphase. Now, what happens during interphase? The cell grows and the DNA is synthesized. DNA is duplicated because the cell will divide. Each cell should get DNA. So the DNA is duplicated. It lasts from 95% of the duration of the cell cycle. 95% of the duration of cell cycle is interphase. Then interphase has three phases. That is G1 phase or gap 1 phase, or antiphase, S or synthetic phase, G2 or gap 2 phase. Okay, so you'll copy this. Okay, and gap 1 synthesis phase and gap 2 phase. Okay. Now let's see what happens during gap 1 phase. So during G1 phase, which is the first growth phase, it is interval between mitosis and DNA replication. So main events is cell continues to grow, cell becomes metabolically active. All metabolism will start. Prepares machinery for DNA replication and synthesis of RNA and protein. So you just write synthesis of RNA and protein during G1. Synthesis of RNA and protein. So G1 is synthesis of RNA and protein. Next is synthesis phase. So in this, you have to know that DNA is duplicated. The DNA replication takes place. Amount of DNA per cell will double. It will become twice. Okay. But chromosome number is not increased. Chromosome number will be same. Only the DNA gets doubled. They are joined. The two DNA will join to each other. Chromatids. In animal cell, replication begins in the nucleus and the centriole duplicates in the cytoplasm. In animal cell, there is one organelle called centriole, which will duplicate. It will go towards the opposite pole. Okay. So from this, you just read DNA replicates. Okay. Why the DNA is becoming doubled here? Why the DNA is doubled? Listen though. What is happening? Cell is about to divide. So two cells will be formed. Each cell should get DNA. So DNA is double. One part will be kept with one cell. The other cell will take the other part of the DNA. Right, only DNA is synthesized. Huh? Now in G2 phase, which is second growth phase, cell growth continues. More synthesis of RNA and proteins continues, and cell is prepared for mitosis. The cell is now prepared for mitosis. So here also you can just write more RNA and proteins are synthesized in G2.
RNA
and protein. Let's go to the next slide. Now, M phase, which is mitotic phase. It has two parts, karyokinesis and cytokinesis. Karyokinesis will occur first, that is division of nucleus, and cytokinesis will occur later, which is called as division of cytoplasm. So it represents the actual cell division. Mitosis is where the cell will actually divide. In human cell, it lasts for only 1 hour. M phase includes karyokinesis, that is nuclear division, and cytokinesis, division of the cytoplasm. So write this last 2 points. Yeah. The next slide. M phase, some cells do not show division. Example, heart cells. Many other cells divide only occasionally to replace the damaged or dead cell. The heart cell never divides after a certain age. And some cells, they divide only for replacement if there is any wound and all. Then M phase, mitosis phase, the cells that do not divide will enter in a phase called G0 stage. Okay. Such cells remains metabolically active, but they do not divide. They do not make copies. Okay. So write G0 phase, write the first point, G0, and then write the cells that do not divide further exits G1 phase. It will not go in G1 phase, it will go in a G0 phase, where it is active. Huh? So it can again enter in a mitotic cycle phase. Okay, G0 phase. Okay. Then second part is mitosis. Okay, which are the 4 stages of mitosis? Prophase, metaphase, anaphase, and telophase. Okay, so mitosis, it is a cell division that occurs in somatic cell, that is non-reproductive cell, the body cell. Okay, it is also called equational division because the chromosome number will remain same. If there are 46 chromosomes in one cell, after mitosis, it will produce 2 cells which will have 46 chromosomes. Okay. Mitosis is generally seen in diploid cell. It also occurs in haploid cell of some lower plants and some social insects. It involves major recognition of all cell components. So write the first 2 points. . Okay, so diploid, what is diploid cell? Diploid means two sets of chromosomes. Okay, 46 means 23 pairs. That is called diploid. Haploid is where it will have only 23 chromosomes. For example, sperm and ovum, it will have 23 chromosomes. It is called as haploid. They will fertilize. Means 23 from sperm, 23 from ovum, fertilize will become how many? 46. You get back the diploid. So here in mitosis, if diploid cell undergo mitosis, the cell which you will get is diploid only. It's also said that some haploid cell also undergo mitosis. So if a haploid cell undergoes mitosis, you will get a haploid cell. Because haploid, diploid depends on chromosome number. Okay. Next is prophase. Okay, so these are the four stages of karyokinesis. That is prophase, metaphase, anaphase, and telophase. Okay, we'll write later. Now, let's see what happens in prophase. The characteristics events during prophase. Early prophase, prophase is the longest phase. Okay, prophase is the longest phase in mitosis. It follows the S and G2 phase of the interphase. After the G2 phase, there will be prophase. In the S and G2 phase, DNA molecules are intertwined. Okay, they are just like a thread, all spread everywhere. Now, in late prophase, chromosome material are untangled and condensed to form mitotic chromosome. So you can see the picture difference. This is how the DNA was. Now they have come together and they have formed chromosomes. They are seen to be composed of two chromatids attached together at centromere. You can see two chromatids attached to the centromere. Okay. Centrosome begins to move towards the opposite pole. In animal cell, the centrosome, which is an organelle which helps in cell division, it will move towards the opposite pole. Each centrosome radiates out microtubule called esters. So they will give out fibers, esters. The two esters together with spindle fiber form mitotic apparatus. Okay. Cells at the end of prophase do not show these structures like Golgi complex, no nuclear membrane or nuclear envelope, no endoplasmic reticulum, and nucleolus. Okay. Next is metaphase. The nuclear envelope is completely degraded now. Okay. Hence, the chromosome can spread. They can move wherever they want. Chromosome contents is complete. Means the chromosome will be all together now. The chromatin fiber will come together and you can clearly see the chromosome under the microscope. They can be studied easily under the microscope. They will have two sister chromatids. You can see, no, two they are. And they are joined at one center called centromere. Chromosome come and lie to the equator. The plane of alignment of chromosome at metaphase is called as metaphase plate. The spindle fibers of both the poles are connected to the chromatids via their kinetochore in the centromere. The spindle fibers, you can see the red one here, red one, they are joined to this structure, see, that is called kinetochore. Next is anaphase. It is the shortest phase in mitosis. Centromere of each chromosome divides longitudinally. Centromere will divide here. Okay. And the two daughter chromatids, chromosomes of the future daughter cell. So you are pulling the chromosome away. A for anaphase, A for away. As the spindle fibers contract, the chromatids move towards the opposite pole. Okay. And last is telophase. Chromosome will cluster at opposite poles. The chromosome will cluster at opposite poles and uncoil into chromatin fibers. Again, it will uncoil. It will become thread-like structure. Nuclear envelope will be formed again. around the chromatin fibers. Thus, two daughter nucleus are formed. Nucleolus, Golgi complex, and ERA reappear. The spindle fiber disappear. So this telophase is always opposite of prophase. Whatever happens in prophase, opposite will be in telophase. In prophase, nucleolus, Golgi complex, and ER they disappear. Whereas here they are reappear. Nuclear envelope disappear. Here they, you can see the nuclear envelope. Okay. Okay, then we have cytokinesis. It is the division of cytoplasm resulting in two daughter cells. It is followed after telophase. Okay. So write one point on each, prophase and all.